Hi, my name is Sushil Kushte and today I am going to discuss one research paper with you. Apoptotic anti-cancer activity of a novel fatty alcohol ester isolated from cultured marine diatom, Phaeodactylum tricornutum. So what are we going to discuss exactly? So our main focus will be on studying various separation methods used during the research and working principle of each method. Let us start with a question. Why did they carry out such research? What was the need? As we all know, cancer is a leading threat for world population and first leading cause of death in the developing countries. Therefore, finding a cancer cure therapeutics is highly demanded. You can see one word written here, apoptosis. Do you know what is apoptosis? If you know, then you can pause this video right now and you can write your answer in comment box. Okay. So apoptosis means programmed cell death and characterized cell changes including blebbing, cell shrinkage, nuclear fragmentation, chromosomal DNA fragmentation, etc. So this research was carried out in Korea and the marine diatom was provided by Korea Marine Microalgae Culture Center. The algae were inoculated in 30-liter plastic cylinder at 20 degrees Celsius after pre-cultivated in 5-liter glass vessel, and the air was continuously supplied by airlift method. We can see the airlift method here. So the microalgae species were cultured in Conway medium prepared from filtered seawater, and it was recovered with centrifugation using basket centrifugation. Then the harvested diatom biomass was frozen at minus 80 degrees Celsius and preserved until freeze dry. So let us first discuss the components of Conway medium and let us also talk about basket centrifugation. So as we can see here, the filtered seawater serves as a base, providing essential salts and trace elements. Sodium nitrate, which is a nitrogen source, sodium orthophosphate, which acts as a phosphorus source, silicic acid, which is necessary for diatom growth, ferric chloride, which provide iron and essential trace element, vitamin and trace element solutions, and micronutrient solutions. So let us talk about basket centrifugation. In basket centrifugation, there are 5 stages. First stage is feeding. So the suspension, that is solid liquid mixture, is fed into the rotating basket, which is perforated to allow liquid to pass through it. Then the next stage is rotation. The basket spins at high speeds, creating a strong centrifugal force. This force acts on the particle in the suspension, causing the denser solids to be flung outwards against the wall of the basket. Then the third stage is filtration. As the basket rotates, the liquid phase, that is filtrate, passes through the perforations in the basket and it is collected outside the basket. The solid phase, that is cake, remains inside, forming a thick layer against the basket walls. The fourth stage is separation. The centrifugal force continues to push the solid particles outwards, compacting them into a dense layer. The filtrate is continuously removed through the perforation. Then fifth stage is discharge. Once the separation is complete, the machine is stopped. The solid cake can be removed either manually or automatically depending on the design of the centrifuge. Now we'll start with the extraction. We can see various methods of extraction here. So let us start with sonication. Okay. So sonication is a technique that uses sound energy, typically between 20 kilohertz to several gigahertz, and it is used for cell lysis. So let us know the working principle of sonication. Ultrasonic wave generation. So this transducer generates ultrasonic waves, then transmission of waves. So we can transmit the waves directly through the probe or indirectly through the water bath. Then cavitation. The ultrasonic waves cause rapid pressure changes in the liquid, leading to the formation of cavities, or we can say microscopic bubbles. Bubble collapse. The bubble grow during the negative pressure phase and collapse during the positive pressure phase. This collapse generates intense localized energy in the form of shock waves. This leads to disruption of cell walls, breaking of intramolecular bind, etc. So the second step, solvent-solvent or liquid-liquid chromatography. Solvent-solvent chromatography, also known as liquid-liquid chromatography, is a technique used to separate compounds based on their differing solubilities in two immiscible liquid phases. This method leverages the partitioning behavior of two analytes between these two phases, typically an aqueous phase and organic solvent phase. So in this case, four different fractions such as N-hexane, chloroform, ethyl acetate and aqueous extract were separated with varying polarity. In that, the N-hexane fraction was found to be active. The third step, solid liquid phase chromatography. The solid liquid phase chromatography is also known as adsorption chromatography, is a technique used to separate components of a mixture based on their differential adsorption onto a solid stationary phase and the solubility in liquid mobile phase. The N-hexane fraction was subjected to solid liquid phase chromatography using hexane and ethyl acetate as solvent with increasing hydrophilic character. 14 fractions were obtained and it was followed by a thin layer chromatography. So thin layer chromatography (TLC) studies were done by staining solvent system and visualized the eluted fractions. So let us talk about thin layer chromatography now. Thin layer chromatography is a simple, quick and cost-effective analytical technique used to separate non-volatile mixture. It operates on a principle of adsorption chromatography where compounds in a mixture are separated based on their differential affinities towards stationary phase and mobile phase. Components of TLC, uh, which are as follows: stationary phase, a thin layer adsorbent material, usually silica gel, alumina, or cellulose, is coated onto a flat, inert substrate like glass, plastic, or maybe aluminum plate. Mobile phase, a solvent or a mixture of solvents that moves up the stationary phase by capillary action. The choice of solvent depends on the nature of the sample and the stationary phase. Among the 14 eluted fractions, F1 and F7 fractions were separated with enough purity. Furthermore, both F1 and F7 fractions were labeled tentatively as PTH1, COM1 and PTH2, COM2 until their molecular structure was determined. Also, F8 was further eluted and further fractionated using preparative thin layer chromatography (PTLC) in, into three subfractions such as PTH8-1 COM3, PTH8-2 COM4 and PTH8-3 COM5. So let us talk about PTLC, that is preparative thin layer chromatography. It is an extension of standard TLC. It allows for the collection of sufficient amount of separated compounds for further analysis or use. So this is how they extracted 5 compounds. You can see these are the 5 compounds. Then the HL60, A549 and B16F10, these cells were grown and these cells were incubated and maintained at atmosphere of 5% CO2 at 37 degrees Celsius. So the cell growth inhibitory activity, that is cytotoxicity, was assessed against the cancer cell lines HL60, B16F10, and A549 using colorimetric MTT assay. So let us talk about colorimetric MTT assay. So this type of colorimetric assay is used for assessing cell metabolic activity. It is widely used in cell biology and drug discovery to measure the the cytotoxicity and viability. The assay is based on the ability of mitochondrial enzymes in metabolically active cells to reduce the yellow tetrazolium salt, that is MTT, to purple formation product, which can be quantitatively measured. So, principles of MTT assay. MTT reduction. MTT is a yellow water soluble compound with viable cells with active metabolism convert MTT into insoluble purple formation via action of mitochondrial dehydrogenase enzyme. Formation solubilization. The insoluble formation product is solubilized using a solvent such as DMSO, isopropanol, or a specific MTT solubilization solution. Quantification. The absorbance of solubilized formation is measured using a spectrophotometer at a wavelength typically of 540 to 600 nanometer. The absorbance value correlates with the number of viable cells. So after assessing the cell metabolic activity, HL60 cells were treated with isolated compound NaMO at concentration of 12.5, 25, 50 and 100 grams per ml and allowed to harvest after 24 hours. So if we take a look at statistics, we can clearly see that compound 2, nonyl 8 acetoxy 6 methyl acetonate, NaMO, was assessed the inhibitory effect of growth of cancer cell lines compared to the control, that is cancer cells without treatment of NaMO. But how can we say that NaMO showed apoptosis? The isolated lipid ester compound at various concentration was incubated with HL60 cells for 48 hours. To determine the apoptosis-induced nuclear morphology of HL60 cells, various treated NaMO concentrations were stained and visualized by fluorescent microscopy. In this experiment, the control, that is non-treated sample, showed the intact cell nuclei without DNA damage. However, during the NaMO treatment, the different concentrations clear DNA damages were observed and led to increase the apoptotic body formations. The isolated fatty alcohol ester from the cultured marine diatom was capable of inhibiting the growth of human leukemia cells significantly. Hence, the functional lipid molecule has the potential to be used in pharmacological and nutraceutical industries, since the cultured marine microalgae could be a good alternative for future bioresources. So this is how a novel fatty alcohol ester was isolated from a marine diatom, Phaeodactylum tricornutum. Thank you.